All right. So again, let me go ahead and back up a little bit. So in order for us to best understand how we perceive the world around us, we need to take a bottom-up approach when we're talking about sensation and eventually perception. And so this means that we need to start examining the smallest components in our sensory and perceptual systems, and then work our way up, so to speak, kind of in this like inverted pyramid, to see how these smaller components are then organized into larger wholes. And so we're going to be talking about visual perception as both an example of really this bottom-up approach to understanding sensation and perception, but also because our visual perceptual system, this is a really core part of cognitive psychology. So we are going to talk about how vision, both in terms of how our eyes take in information and how our brain processes it, how vision happens. And like I said, we'll be talking about that to not only show an example of this sort of bottom-up processing approach, but also to help you become better familiarized with vision. So let's go ahead and review the basic anatomy of the eyes and the brain's visual processing system. So again, we're going back to Gen Psych information. So if you remember, the building block of the neural system is called a neuron. And so neurons are specialized cells that occur mainly in the central nervous system. And the central nervous system is comprised of the brain and then the spinal cord. So it really lives up to its name. It's the nerves that are in the center of your body. And so, you know, neurons, they vary to some degree in terms of their structure, but generally they all have this sort of uniform setup that we'll go through. So they're made up of what's called a soma, or the cell body, dendrites, an axon, and terminal buttons. And so the soma forms the body or the cell body of the neuron. And this is what contains the nucleus of the cell. And so what the cell body does is it adds information together that it receives from the dendrites, which we'll talk about in a moment. And then it passes on this collection of information down to the axon. And so for the dendrites, these are the sort of branch-like or like tree-like structures that extend from the soma or the cell body. And so what the dendrites are doing is they are collecting information from hundreds of the other neurons that are connected to them. You can't see it in this picture, but there's really this ever expanding collection of neurons that are connected to each other. And the dendrites then take the information that they get from other neurons and they pass it along to the cell body. And then from the cell body, information is passed on through the axon. And so the axon is, there we go. The axon is this long, slender tube that extends from the other end of the soma from the dendrites. And when it's signaled, it sends this electrochemical signal down its length to the end of it, which results in more of these branches that are called terminal buttons. And some axons, not all, but some are covered in this white fatty substance called myelin. And because it's wrapped around the axon, it's called a myelin sheath. So it's It's wrapped around the outside of it. And as you can notice in this picture, in this diagram, the myelin sheath isn't just one continuous structure, but it actually appears in segments. And the gaps or the spaces in between these segments of myelin are called the nodes of Ranvier, which sounds really fancy. And actually, the inclusion of this myelin sheath and the nodes of Ranvier actually allow the message to be passed down the axon really quickly, even more quickly than it would if it was just one continuous myelin sheath. And the reason for this is because the signal can then jump from node to node. So from node of Ranvier to node of Ranvier. So the way I kind of think about it is the axon being like a sidewalk. And if you didn't have that myelin, you could just walk down the street. But the nodes of Ranvier kind of introduce almost like this pogo stick quality of information transfer. And so you're going to get down the sidewalk a lot faster if you have a pogo stick. You can take way bigger bounds with a pogo stick than you can with your own feet. I mean, unless you're somebody who's incredibly tall, but I don't know anything about that life. So I'm sticking with my pogo stick as something that I think will outpace regular walking in this metaphor. And then we move on to the final structure that we'll talk about. And so this is the terminal buttons. And so these are these sort of little knobby ends at the end of the branch-like extensions that come off of the axon. And the terminal buttons and the dendrites of the next neuron that they connect to, they actually don't touch in this connection. There's actually a small gap of cellular fluid, and this gap is called a synaptic gap or cleft. You've also heard it called a synapse as well. And so what the terminal buttons do is they release a chemical called a neurotransmitter into the synaptic gap or the synapse. And then the neurotransmitter then floats across the fluid from one neuron to another. And then it locks onto the receiving dendrite. And then the dendrite takes this neurotransmitter chemical and then passes it along as an electrical signal onto then its own cell body. And as you might know, the job of a neuron at its most basic is to communicate with other neurons, obviously, talking about all of this. And as I referenced earlier, they do this electrochemically. So neurons are using both electrical and chemical forms to communicate with one another. And neurons are also typically organized in networks, and this allows them to pass information back and forth along established pathways, which we very frequently refer to as networks. So based on that quick overview, let's then talk about how we see. So we'll start with the structure of the eye and what happens there, and then eventually we'll move along to how our brains make sense of the visual information that comes into the eye. So our eyes have a variety of structures that allow us to process and send along visual information. And so again, this might be review depending on classes that you've been in before. But real quick, the highlights of it are that light enters our eyes through the cornea, which is the very first layer of the eye, after which point it passes through the pupil, which is the black portion of the eye that dilates and constricts. And then it goes through the aqueous humor, so this fluid, and then it goes through the lens, if you've ever known anybody who has had cataract surgery, the lens is actually the part that is affected by the cataract, and that is what is surgically replaced. And then after the lens, it goes through the vitreous humor. So this is the fluid that really gives our eye its spherical shape and gives us the sort of pressure that we need to keep it shape. And then the light eventually hits the back of the eye, which is called the retina. And oh, another thing about the lens too is you've likely seen like diagrams where the images that are entering our eye are then flipped when they hit the retina. And the lens is the structure that actually inverts or flips that image. And so the retina is made up of these specialized cells called photoreceptors. And there are two types. You've probably heard of them, rods and cones. And rods, these are what specialize in detecting lower levels of light and are how we're able to perceive just sort of general distinctions between objects when there's less light that's available. So our rods are coming into play when you wake up in the middle of the night and you see what you think is somebody in your room, and then you realize, oh, wait, no, that's actually just a pile of clothes that I left on my chair or on my desk. That's not an intruder. So the rods are what are helping you distinguish between the various shapes that it's picking up on in the low lighting of your room. And so the rod receptor cells, these are more prevalent in the outer parts of the retina. And whoever named them was not creative. They just called them rods because they look like rods. I'll skip ahead. This is a picture. On the left, you have a rod. On the right, you have the other photoreceptor type, which is the cone. So not a lot of creativity on there. Whoever discovered them was like, we'll keep it real simple. And so cones then are the photoreceptor cells that then allow for color vision. And there are three different types of cones. There are blue, green, and red. And so they're named this because it corresponds to the wavelength, the frequency of light that they respond to. And cones are concentrated in the center of the retina. So the very center of the retina is called the fovea. And the concentration of the cones in the fovea is what allows us to perceive really fine details. And again, like I said, they are called cones because they look like cones. And so the light that, when it finally reaches the retina, it then triggers a chemical reaction in these rods and cones. And then this is converted into an electrical signal. So again, going back to our just general understanding of neurons, we have, again, this electrochemical communication system going on here. And so the rods and cones then transmit an electrical signal, and it's amplified across the various layers of the retina. And then it's eventually sent to the brain for further processing via the optic nerve that comes out of the back of the eye. Interestingly enough, so because of where the optic nerve exits the retina, it results in what's called a blind spot because that nerve tissue is actually taking up the space where rods and cones would be. So there aren't any photoreceptors there. So that tissue is not processing any light at all. Okay, as we talked about the structure. Oh, this is a really cool GIF I found actually on the LensCrafter website that I found was really helpful, just simplified breakdown of all of these very complex steps. Okay, so moving on to talk about frogs and cognition, and hopefully this will make a little bit more sense as we go on. So in the 1950s, there were some researchers who began to investigate exactly how these sensory cells in our retinas process visual information. So the basic driving question that they had was, how do these light dark detectors, rods, and color detectors, cones, how do these really basic simplistic processes then lead to the eventual generation of the detailed visual information that we can pick up on? You know, we're not seeing in black and white, we're seeing in, colors included in there as well. We have this really sort of high resolution visual ability. So how are we going from just determining shadows and colors to having this really rich visual system? And so the researchers at this time, they discovered that each of the neurons that are in the various layers of the retina, they quote unquote cover a particular area in the field of vision. And so this area came to be called a receptive field, as this is where the cell receives the sensory information. And this sensory information then leads to activity or lack of activity in the cell. And so in addition to the photoreceptor cells of rods and cones, researchers also found that there are other cells in the retina, and they call these bipolar cells. And the reason for this is bipolar cells, rather than having dendrites on one end and then an axon on the other, they actually have axons on both ends. And the bipolar cells are connected to the photoreceptors and are kind of the sort of middleman between the photoreceptors and then the higher, more sophisticated cells that we'll talk about. And researchers during this time also found that there are two different categories of bipolar cells. And so, again, they went with just very literal definitions of these things. So the first type they called on-center, or it's more thoroughly on-center, off-surround. And so these on-center bipolar cells respond or fire when light hits the center of their receptive field, so on center, and they don't fire when the area surrounding the center is hit by the light. So they turn on when the center is hit by light, and they turn off when the surround is hit by light. So pretty straightforward name. The other type is the inverse. And so these are called off-center, on-surround. And so, as you can imagine, with these ones, when the light strikes the center of the cell, it turns off the cell activity. Whereas light that falls on the surrounding area outside of the receptive field is what causes cells to respond. And so the result of these on and off center bipolar cells is the construction of edges and lines. It's really basic edges and lines, but this does support our ability to recognize patterns, to recognize the general shape of objects. And so how do frogs fit into all of this? So a set of researchers, Letvin et al., in 1959, they published this really classic article, and it's called "What the Frog's Eye Tells the Frog's Brain." Again, very literal titles here. And so they found that in the frogs that they were studying, that their visual pathways responded to or became active in the presence of different kinds of objects and events. And these researchers came to understand this to mean that there were groups of neurons that would respond to a particular pattern of light as it hit the retina. And one such type of this specialized neuron they called the bug detector, as these were the neurons that would respond when there's any sort of small zigzagging pattern that the eye was picking up. So like that of a fly. And so this research not only told us that there are certain patterns that our neurons will respond to, but that the patterns seem to be tied to the specifics of that organism. So again, this bug detector, frogs primarily eat insects, and so it makes sense that they would have neurons that were specially adapted towards being able to see and track insects. And so following this frog research, cats ended up getting involved too. And so researchers Hubel and Wiesel found that there were specific neurons in a cat's visual cortex, so the visual part of the brain, that would respond when a column of light was flashed on the retina at a certain angle, and also at that angle when it would move. And so these researchers eventually came to call these complex cells. And this was in comparison to the simple cells of the on and off center bipolar cells. So we're starting to see this sort of bottom-up approach of we have these simple cells that then eventually lead to organizations of more complex cells. And these researchers also found that complex cells can then be organized into larger and even more complex cells called hypercomplex cells. Again, not a lot of creativity here with the name, but at least it's straightforward and you know what it's talking about. And so these hypercomplex cells are formed when the axons from several complex cells that have different line orientations that they respond to, and that have visual fields that are close to one another. When the complex cells, what they call converge or attach to their axons to a single neuron. And hypercomplex cells provide even more detailed visual processing for us. So various networks of hypercomplex cells are able to respond to the presence of certain angles, particular moving shapes, lines of a certain length, or even the particular ratio of light to dark in a space. Hubel and Wiesel also discovered that there was further visual organization within the brain's visual cortex, and they termed these hypercolumns. And so these hypercolumns are made up of several components. So one of them are components that are responsible for color sensitivity, so color processing, picking up on the gradient and hues of colors. They call these blobs. Also within these hypercolumns, so this organization of cells within the brain that are involved in visual processing, there are cells called ocular dominance columns. And so these are the cells that specifically allow us to have binocular vision, so being able to see out of two eyes and then integrate that information. And then the final piece of these hypercolumns, these stratified columns of neurons in the brain, are these line orientation detectors. that they just simply called orientation column. So I know that that was a lot of details and jargon, but I just want to summarize and say that overall, the takeaway from all of these details is that researchers, they really began to unravel the mystery of the complexity of vision. So they were starting to be able to understand better the various bits and pieces. So like we see light detection, color processing, motion detection, the detection of edges between an object and what's in the background of it. And these researchers also were able to document the hierarchical organization of the components of the visual system. And so again, this demonstrated that higher and more complex processes were made up of the cumulative effect or the additive effect and organization of the more simple processes. So we're going to move on then and talk about the visual pathways. So essentially, the structures of the brain and processes of the brain that then carry visual information from the eyes to then be processed, to become what we would call perceived. So the resulting visual information from the retina is carried to the brain via the optic nerve. And then this splits into two different directions at a structure called the optic chiasm. The optic chiasm is this X shape. Chi is Greek for X. So this X-shaped structure in the brain. And again, drawing back on previous classes, you may or may not remember that our brains are organized in such a way that each hemisphere, each half of the brain controls the opposite, or the more technical term for this is contralateral side of the body. So this means that the right hemisphere controls the left side of the body, and the left hemisphere controls the right side of the body. And so if we just take a look at this really simple diagram here, and specifically how the eyes are positioned, it's hard to imagine how the right eye sends information to the left side of the brain and the left eye sends it to the right side of the brain, because we are seeing this whole integrated field. It's not like, there's some sort of divider between our eyes, or if we're, walking around with one eye shut, going, oh, okay, well, now my right eye is picking up things in the right side of the field, so I guess it's sending it to the left side, and like vice versa over here. No, actually, both of our eyes are having to integrate this information, but then send it to the appropriate processing centers on the appropriate side of the brain. And so to illustrate this, for me, at least looking at these sort of static diagrams of how this happens has actually been pretty confusing for me. And so I want to show a video that I think really nicely illustrates how the visual information gets sent to the contralateral areas of the brain for processing. Okay, so sorry, let me make sure that it is showing the right. Here we go. Okay, is everyone able to see a YouTube video in full screen?
Yes.
Wonderful. Thank you, Madison. Okay, so we're gonna go ahead and watch this video. Again, for me, this really helped me make sense of the more convoluted charts of vision that you'll see in textbooks.
In most of our body, we have the right side of the body being controlled by the left side of the brain, and the left side of the body is controlled by the right side of the brain. So how does this work in vision? So let's imagine that this rectangle that I drew is our entire visual field. And if these two eyeballs were focused in at the center of this rectangle, so if they were both focused in on this purple line, all they can see are these two colors. So we have a ray of light coming in from the left visual field. It'll hit the eyeball, it'll hit the left eyeball, it'll kind of be bent a little bit by the lens, and it'll hit the right side of this eyeball. And so the inner side of the eyeball, so this side of the left eyeball and this side of the right eyeball is known as the nasal side, because the nose would be right in the middle of the eyes. So the nasal side is the side of the eyeball closest to the nose, and then this outside part of the eyeball, which is over here and over here, is known as the temporal side of the eyeball because it's closest to your temples. So this is the side closest to the temples, this is the side closest to the nose. So a ray of light coming from the left visual field will hit the nasal side of the left eye, and a ray of light coming from the left visual field will hit the right eye, be bent a little bit by the lens, and it'll hit the temporal side of the right eye. So let's look at a ray of light coming from the right side of the visual field. A ray of light coming from here would enter the right eye, and it would be bent a little by the lens and hit the nasal side of the right eye, whereas a ray of light coming in would hit the left eye, be bent a little, and hit the temporal side of the left eye. So let's look at what happens next. So the eye is basically connected to the brain via the optic nerve. So there's an optic nerve that kind of exits the back of the eye and goes into the brain. So interestingly, the optic nerve from both eyes actually converge. So they actually reach a point where they converge. And this point right here where they converge is known as the optic chiasm. So they kind of converge and then break off again and then move even deeper into the brain. So this point where they converge is known as the optic chiasm. So optic chiasm. Let's look at how this information is transmitted to the brain through the optic chiasm. The retina is lining the back of the eyeball, and we had this yellow ray of light hit the nasal side of the left eye, and it hit the temporal side of the right eye. So let's go ahead and trace this information to the brain. So the information will be sent via axons through the back of the eye into the optic nerve, and basically it'll come in, and what it'll do is it'll actually cross at the optic chiasm and then go this way. And what we also have is this ray of light will come in to the back of the eye, and it will actually go down the optic nerve, but it's not going to cross. So all light that hits the temporal side of either eyeball does not cross the optic chiasm. So let's go ahead and trace this green ray. So the green ray coming from the temporal side of the left eye is going to exit the back of the eye, go down the optic nerve, and it's just going to stay in place. So it's not going to cross the optic chiasm, whereas this ray of light is going to go through the back of the eye, and it's going to cross over here. It's going to cross the optic chiasm and go down to the brain. And so what this effectively does is it actually takes the right visual field and allows all the information that's entering the eye from the right visual field to go to the left side of the brain. So this is the left side of the brain. This is the right side of the brain. So like the rest of the body, all the information coming from the right visual field actually goes to the left side of the brain, and all the information coming from the left visual field goes to the right side of the brain.
Does anybody have any questions about that before we move on? Cool. Okay. Well, I just wanted to check in about that because for me, the visual system has actually been like one of the hardest, most detailed things for me to learn about in my graduate program, and like at the neuroscience level, it is brutal. So I'm hoping that this is just a really helpful, bird's eye view of it. Okay, so let's continue on with our PowerPoint here. Okay, so as we saw in the video, so the visual information, it travels away from the optic chiasm, and then it eventually goes back towards the brain. Let me go back to our previous slide. After it crosses the optic chiasm, these nerve fibers are called the optic tract. So before they come to the chiasm, they're nerves. And then after they cross the chiasm, they are called the optic tract. And so this information eventually reaches the occipital lobe. And so this is the lobe at the very back of the brain. And this houses our primary visual cortex. So this is the main area where visual processing is taking place. And so then from here, from the occipital lobe and the primary visual cortex, the information is then sent to other brain areas for further refinement and processing of the details of visual information. So there are two different pathways or streams that this information can travel along. So one is called the dorsal pathway. And so just to orient ourselves anatomically, dorsal, like you can think of like a dorsal fin, right? That's going to be sticking up at the top. And so the dorsal path is going to go up and over the top portion of the brain. And it heads towards the parietal lobe. And the dorsal pathway is also called the where pathway. This is my personal trick for keeping the dorsal and the ventral pathways separate and keeping in mind what they do. So D is for dorsal, and D is also for donde, and donde is where in Spanish. So dorsal, donde, dorsal is where. Great. Okay, dorsal is the where pathway. And so what this means is that the parietal lobe is processing the various aspects of visual input that pertain to spatial location, the shape of something, sort of its vertical or horizontal orientation. And it also gives us information that helps us with reaching and guiding and grasping objects as well. So the dorsal pathway really helps us to navigate our environment as it is in space. And information can also travel along the ventral pathway. So this is the what pathway. And so the ventral pathway goes down along the bottom side, more bottom side of the brain. And it heads towards the temporal lobe. And so then here, processing that takes place is at the level of recognition and being able to discriminate visual shapes and objects from one another. So as an example, the dorsal stream is what allows me to determine exactly where my water bottle is on my desk. Whereas the ventral stream is what allows me to recognize it, as a water bottle. So I can look at it and I can name it, say, oh, that's a water bottle. And the ventral stream also allows me to distinguish it from the other objects on my desk. So if I was thirsty, I would know that I should grab the water bottle rather than the cup of coffee that's next to it. Well, I guess if I was thirsty, I would drink the coffee too, but really trying to stay hydrated these days. So in this particular example, I would be able to differentiate and recognize, oh, this is a water bottle. This is the one that I want. Okay, so that was a lot of information, but again, it was in the service of not only understanding just sort of the basics of human visual sensation and perception, but also to give us this idea of a bottom-up approach. And so, you know, these physiological properties and components of vision have shown us that the simple sensory data or information that we get can then be added together to make up more complex processes or ideas in this sort of increasingly hierarchical organization. And so with a bottom-up approach, we can see that each higher process or level can be described as just being made up of the sum of what came before it. So as an example, the hypercomplex cells that Hubel and Wiesel described, the hypercomplex cells are then made up of complex cells, and complex cells are made up of simple cells. And so this kind of, yes.
Sorry, I think, Chase, did you have a question?
Yeah, I did. So
That's okay. Yeah. Sorry, I didn't see it, Chase.
No, you're good. For I think it was the ventricle system that goes down. Or
the ventral system? Yeah.
The ventral system. If you don't recognize something, like if you can't actually differentiate it, what happens then? If that makes sense. Like, does it go back to somewhere else where now you just know that there's like, can it go from like one place back up to maybe just like, okay, well, we just know where it is, but we don't know what it is?
Yeah, great question. And so we know that, you know, our cognition is really complex. And so this is obviously a really simplified diagram, but this information is actually being sent in both directions because we need an integration of this information too. So to recognize, you know, my water bottle and to be able to grab it, I'm actually combining my understanding of where it is and what it is at the same time. So it's not necessarily that, you know, the dorsal stream is firing and then it's figuring out the, you know, spatial information, and then the occipital lobe is like, oh, okay, now we'll fire the ventral stream. But they're happening together sort of in tandem simultaneously. And in terms of your question about, you know, if there was some sort of breakdown or issue in one of the streams, that's great. We will actually be talking about conditions that can result from brain damage, from things like stroke or tumors or traumatic brain injury, called agnosias. And so essentially, not to get too detailed at this point, but in this example, so say that, you know, there's damage in the temporal lobe, in some of the structures that are involved in the ventral stream, so the what processing, there's going to be a communication breakdown that happens there because the networks in that area, the collection of cells, have been disrupted because of the damage. So with agnosias, you can have difficulty recognizing certain objects. You can also have a specific kind of agnosia called prosopagnosia, which is also called like face blindness, where you are unable to differentiate the faces of people from each other, even if it's somebody like your wife or your mom, your brother. And so, yeah, there are disruptions that can happen along the paths not just of our senses, right? So you can have damage to your eye that then is going to impede information being sent to the brain. But then also at the brain level, you can have these breakdowns in communication that then impact how we perceive and understand the world too. So does that make a little bit more sense, Chase?
Yes, thank you.
Okay, awesome. Cool. Yeah, and so if you ever have a question and I'm not getting to you, you can just pop in and interrupt me. So the way that Zoom does it when I'm sharing my screen is I just have like a little tiny box. And sometimes I'm able to see that someone is like raising their hand, but other times I can't. So just an FYI, please know I'm not ignoring you. I just don't see you. I'm sorry. Okay. So moving on from this sort of bottom-up approach that we've been taking, a bottom-up approach is also sometimes referred to as being reductionist. And if you are philosophical, you might have heard this term. And so a reductionist kind of perspective is this idea that there's an assumption that a whole something, an object, a person, etc., can actually be reduced down to its component parts. So in opposition to this, we have the field of Gestalt psychology. And Gestalt psychology embodies what is called a top-down understanding of perception. If you've ever heard the phrase before, the whole is greater than the sum of its parts. This really illustrates the Gestalt position well. There is a Gestalt psychologist named Max Wertheimer, and he wrote this quote in one of his papers, which I think is great and really kind of sums up this idea. So he wrote, there are wholes, the behavior of which is not determined by that of their individual elements, but where the part processes are themselves determined by the intrinsic nature of the whole. So essentially, we have this focus on the whole object itself rather than really getting into the nitty-gritty of the various parts of it. And so the reason why we are talking about Gestalt psychology in the context of sensation and perception is that this particular field has been influential in cognitive psychology. It's not an extensive sort of field per se, but it has brought insights to cognitive psychology in terms of perception that have been really important and have really informed us and have also helped us to take not only a bottom-up perspective to understanding, well, essentially understanding, but also a top-down approach as well. And so there are some popular Gestalt concepts. You'll probably recognize some of them, or you probably did recognize some of them when you were reading the book. So the first one is called it's called the phi phenomenon. And so this is a sort of optical illusion in which you, the viewer, perceive that there's movement happening, but actually there isn't movement happening. It's just the result of when fixed objects like light bulbs or a certain set of pictures are lit or shown and then covered up one after the other in quick succession. So an example of this here, so on the left, it looks like these dots are moving, like there's some sort of movement that's happening here. But what's really happening is there are just frames that are being transitioned between where each of the dots is being just blocked out. So it's not that the dots are moving actually, but that's how our mind is perceiving it and making sense of it. It's saying, oh, there's movement that's happening there. Same with like this like buffering icon. Again, it looks like, oh, okay, you know, the wheel's spinning, but it's actually not spinning. It's just the result of different frames that have different objects and different orientations of them. So like if you've ever seen like a comic like Flip Book, our brains are integrating that so that it looks like it's just smooth movement. We're not picking up on the individual sort of frames or pieces of this, but rather we're seeing it as one moving whole. And so another concept that we've gotten from Gestalt psychology is the relationship between figure and ground. And so this is when our visual systems tend to focus in on the main object that we're looking at, so the figure, and then everything else becomes then relegated to the background. So a fun example of this are if you've ever seen pictures like this. So on the left here, depending on what you are looking at, so depending on what your mind is defining as the figure, you might see a tree, a black tree, and then a white background, or you might see a gorilla and a lion with a black background. So similar, this one on the right is a really famous instance of this. So depending on what your brain is defining as the figure and what it's defining as the background, you are either going to see a black vase or two white silhouettes that are sort of looking at each other or across from each other. And so Gestalt psychologists, they propose that there are certain organizational factors. So there are certain sort of rules or laws that guide our perception. So one such rule is called the factor of proximity. And so this is our brain's tendency to group items that are close together into a particular collection or a shape. So here we can see that the black and orange dots, we could group them based on their color, but here, the idea of proximity, so these two groups being close together, making one whole, and these two groups over on the right together, sort of making another whole, it actually is how our brain is organizing them. So the idea of proximity, so how close things are gathered together in our visual field, often overrides other ways that we can distinguish between stimuli. So for example, here, we're not really seeing, oh, well, there's, you know, this orange sort of shape and then these two black dots. We're seeing, nope, here is one set of dots and then here's another set of dots. And so another organizational factor is the factor of similarity. Sorry, I'm having to switch back and forth. I was going to try to get fancy, and then I just didn't have time. So I apologize about that. So like I said, another factor is called the factor of similarity. And so this says that objects that are similar to one another will be grouped together. So here in figure A, we have similarity of color. So here we have the similar dots, the gray dots, are grouped into sort of like one object that's distinct from the rest of them. Here in figure B, we have similarity at work. And in this case, it's the similarity of line orientation. And so the squares that are made up of two vertical bars actually appear as one shape. And as we can see, these two bars our brain sees as similar. And then the orientation of the lines over here makes this appear as a dissimilar sort of object. And so again, we like to group things based on how similar they are to one another. And so another fun factor that our brains like to use when we're organizing things in our visual field is this idea of global versus local organization. And so this says that the global, or the sort of larger or more macro features of an object is what is going to dictate, there we go, how we are organizing things versus the local or smaller characteristics. So here, rather than seeing, oh, okay, well, this is, you know, a line of F's here that is then, you know, intersected by two more F's, and then we have another F here. You see this as a whole. You see its global shape of an H. Same thing over here, rather than parsing this out as, oh, okay, well, you know, there's a set of L's and then these ones run down. No, you see the whole global shape. It's an F. And then the last big factor is called the factor of good continuance. It's sometimes also called the factor of closure. And so this is our mind's tendency to close lines or even continue lines that are actually not present. And so these are called Kaneza triangles. And so as you can see that when you look at them, you think, oh yeah, like there are two fully closed triangles. But then if you look closer, you can actually see, well, this triangle is actually not made up of any lines. It's just blank space. And the sort of triangle that appears in the background or underneath it, it's actually these lines. We don't actually know if these lines are connecting or not. Our mind thinks they are, yeah, and underneath this triangle. But when we look, these lines aren't actually connecting into a triangular shape. But because our brain likes to close things, complete patterns, and make meaning out of them in that way, we have this idea, oh, there are actually two overlapping triangles here. Okay, so I know that we have just a few more minutes, so I'm going to go ahead and stop there and just check in and see if anybody has any questions.